Hi, welcome to Rocky Biochemistry series. In today's video, we will be discussing the Lac Operon. If this is your first time of watching our video, kindly subscribe for more important videos of some key topics in biochemistry and molecular biology. Today we will look into how genes are regulated in bacteria, how bacteria toggle on and off the transcription of some specific genes, and how the bacteria is able to recognize environmental conditions in which it should activate or repress the expression of some specific genes. So an operon is just a segment of DNA that encodes for a multigenic mRNA. In bacteria, they are nutritional opportunists, meaning they give priority to the kind of nutrients that they should metabolize. So for instance, when there is glucose and lactose in the medium, preference will be given to the glucose, and the lactose will only be metabolized when there is no glucose in the medium. In order for the bacteria not to waste energy in transcribing the genes for the metabolism of the lactose, it shuts those genes down when there is glucose in the medium. And such genes are only activated, or such genes are only transcribed when there is no glucose in the medium. So the Lac Operon is made up of, we have the Lac I gene, which encodes for the repressor, the Lac P, which encodes for the promoter, the Lac O, which encodes for the operator, and then Lac Y, which encodes for the permease, the Lac Z, which encodes for the beta-galactosidase enzyme, and the Lac A, which encodes for the trans-acetylase. So the repressor is actually not part of the Lac Operon. The repressor is actually not part of the Lac Operon, but the Lac Operon is just made up of the promoter, the operator, the permease, the beta-galactosidase, and then the trans-acetylase. The Lac I gene, as I said earlier on, encodes for the repressor, and then Lac P encodes for the promoter, which serves as the binding site of the RNA pool, and then the O, that's the operator, serves as the binding site of the repressor, and then the permease enzyme is responsible for the transport of lactose from outside of the medium, or from the outside of the cell to the inside of the cell. And then the beta-galactosidase is responsible for the cleavage of lactose into galactose and then glucose, for the glucose to be metabolized. And then the trans-acetylase inherently plays no role in the metabolism of lactose. So let's look into when there is no lactose in the cell. So when there is no lactose in the cell, what happens to the Lac Operon? The Lac I gene, as usual, will encode for the repressor protein. And after encoding for the repressor will bind to the operator region. Upon binding, the RNA pool will also bind to the promoter region. But due to the presence of the repressor at the operator region, it blocks transcription of the gene by the RNA pool. Hence, transcription of the Lac genes do not occur when there is no lactose in the cell. Let's look into what happens when there is lactose in the cell. So when there is lactose in the cell, the Lac I gene will as usual encode for the Lac repressor, but the lactose in the cell will bind to the allosteric site of the repressor. And upon binding to the allosteric site of the repressor is now prevented from binding to the operator region of the Lac genes. Then the RNA pool will bind to the promoter region and carry out transcription of the Lac genes for the metabolism of our lactose. Thank you for watching our video.